Okay, so good morning. This is Dr. Correa again. I haven't seen you since August. We're doing small groups next week and the week after. Next week we're doing sickle cell. Because we're doing this online, I guess we're using something called Teams. I decided to reintroduce a PowerPoint that I used to present years ago when we did these small groups earlier in the semester, almost at the beginning of the second block. And so this is meant to be some background material that will be helpful for your self-study on sickle cell, thalassemias, and hemoglobinopathies in general. I've tried to make this PowerPoint a couple of times, and I keep running into some technical issues. This, I think, is actually the fifth today. I had to truncate it yesterday because of problems with slides. You can already see I have some trouble with some other slides ahead, but it's minor at this point. Okay, so your primary source should be Lippincott's. Chapter 3 has a nice discussion of hemoglobin structure function. There's also a really nice discussion on hemoglobinopathies, sickle cell, thalassemias in general. So you should be well aware of what it means to have an alpha or a beta thalassemia, in addition to what sickle cell mutation, hemoglobin S, or the C mutation, or the hemoglobin SC mutation means. A lot of my information comes out of this chapter on hemoglobinopathies in an online textbook. It's actually just a chapter, a book of chapters. Online metabolic and molecular, I think, is the order basis of inherited diseases. This used to be in the library. It's no longer available in the library. They don't want to pay the fee. There's a newer version of this available out there, but you have to basically buy a subscription. But in any event, I have posted 181 in the block under sickle cell groups. I have also posted this chapter, or this book, publication, Journal of Clinical Investigations from 2007. It's a different introduction to the general topic. It's got some historical value, and I'll show you some slides out of that one as well. Okay. So the learning objectives for this today are that you should review and know all the features of hemoglobin structure function and its transport of oxygen from the lungs to the tissues. You should know the cooperativity characteristics of hemoglobin. It binds oxygen cooperatively. And then you should know that factors like DPG and CO2 and protons are low pH, induce right shifts and the binding curve and improve oxygen delivery. You should know what carbonic anhydrase does. You should know some general features about red blood cells. We have already talked about hemoglobin S and C and SC, but we're going to go over that again. And you should understand the inheritance and clinical manifestations of these diseases. This is basically what you really are going to be talking about in the clinical. I will only discuss some of that, but a lot of what you need to do will involve your group private research. In general, you need to understand how hemoglobin mutations might affect function. Specifically, you know that there's a proximal distal histidine that is involved in oxygen binding, and so you need to basically know that. But you know that there's also some histidines that are involved in binding the Bohr proton and establishing salt links that stabilize the D state, or the low affinity state, the deoxy state, or the low affinity state, or the T conformational form. So you appreciate some features about tetramer stability and what causes this conversion between R and T, and how oxygen affinity is associated with that. But you should also then appreciate that these features have impact potentially on the oxidation state of the iron and its ability to bind oxygen. And then potentially mutations might affect what we call solubility. In the case of hemoglobin S, this is coupled to the formation of a large 14-stranded aggregate. So that is what we call an insoluble form that distorts the red blood cell structure. But there are other potential mutations that might disrupt structure, that might disrupt its solubility, and therefore might disrupt tetramer stability or oxygen transport. And we'll discuss a lot of these things in a cursory way as I go through this PowerPoint. And then you need to look again at the gene structure, the hemoglobin genes, how many, where they are, how they're organized. And particularly, we're going to be talking about thalassemias that occur because of alpha hemoglobin deletions or beta hemoglobin mutations that affect mRNA processing. Basically, they tend to affect splicing. And so you either have a loss of genes in alpha or you have a change in protein structure in the case of betas. Okay, and here we go. So you need to basically have this overview, and I'm not going to go through all of this, but basically, where is hemoglobin found? What's its function in terms of oxygen transport, lungs to tissues, and at the same time, CO2 and acid back from the tissues to the lungs, and what the overall capacity is. When we have anemia, we have less concentration of hemoglobin. So this is the general feature that you should sort of have in your mind, that we have this binding curve. Here's a 0 to 1 fractional saturation. This means 4 oxygens bound, plotted versus the concentration of oxygen and partial pressure. Around 100 in the lungs, the hemoglobin is saturated, and around 20 in the tissues, we have delivery of oxygen. There's a right shift in the presence of these allosteric effectors. So things like 2,3-BPG and CO2 binding and high proton concentration, low pH concentration. So you could at the same time write this as low pH, which means higher acid. That would cause this right shift and improve delivery. But it also means that you get an increase in the deoxy form. And it's the deoxy form that aggregates in sickle cell anemia. So this is a general gene structure. And there's a depiction here on the left showing you the organization of the genes. So for example, the beta genes are all on chromosome 11. There's a embryonic form, 2 fetal forms, and then 2 adult forms, a minor delta and a major beta. And you know that the embryonic form goes away very quickly after conception, and the fetal forms come up, and then they go away after birth. Then the adult forms appear, the delta form, which is also known as A2, and the beta form, which is known as A1. And so this expression is in the order of the genes. And then the alphas are similar. There's an embryonic form, I call this symbol psi, and then there are 2 alpha forms called alpha that come up early during fetal development, and then those are the ones that persist. This organization, it turns out, organizes the genes in their order of expression. So they get expressed in this order temporally. And that is controlled by other loci upstream from the genes that control this and regulate this. So for example, they'll turn on fetal hemoglobin at some point, and then they'll turn off fetal hemoglobin. And it's these loci that control that. And we are thinking about and working on mechanisms for changing that modification in those loci so that we can turn on fetal hemoglobin in some systematic genetic way to combat things like sickle cell. We'll talk about that at the end of this PowerPoint. Okay, so this is another summary depiction of that. The adult form, the major form is A2, and so this is alpha 2, beta 2. A minor adult form is, excuse me, this is A1, alpha 2, beta 2, and then A2 is the minor form, usually around 2 to 5%, and this is alpha 2, delta 2. We still express lower levels of fetal hemoglobin, UNI, alpha 2, gamma 2. Some people express higher amounts of this, and we try to induce this, for example. And that's one of the things we'll be talking about in the clinics. And then there are embryonic forms, and this is showing you what those Greek symbols are for those gene combinations. In this different depiction down below out of your textbook, there's also a mention of hemoglobin A1C, which is the non-enzymatic glucose modification of hemoglobin. This is a measure of your general sugar state, how much sugar you have in your bloodstream. And this is essentially a marker for diabetes that we'll talk about in a couple of weeks. So here's another way of depicting these genes, again out of Stryer. Here's the embryonic alpha chain, and then the 2 adult forms. Here's the embryonic beta chain, the fetal beta chains, and the 2 adult forms, and the various combinations of tetramers that you can make out of these combinations in some kind of a temporal way. Since there's one beta chain on this chromosome and we are diploids, there are 2 copies that you can potentially inherit from your mother or your father, and so that impacts heredity, but it also impacts other kinds of genetic modifications and what you might inherit. Since there are 2, so there's a single copy of beta, and that means in the diploid sense, there are 2 copies that we carry around in our chromosomes. There are 2 alpha chains on each DNA strand, and therefore, since we're diploids, there are 4 total alpha genes, and that has implications for disease states for alpha that we'll talk about when we start talking about thalassemias at the end of this PowerPoint. Okay, so in general, the features that we're talking about is that sickle cell is basically an inherited anemia. The sickle cell characteristic destabilizes red blood cell membranes and destroys red blood cells, and so people with sickle cell disease tend to have low red blood cell, low hemoglobin concentrations. Polycythemia is the inverse. It's an increase in red blood cells. It's an increase in general production of cells that come out of the bone marrow. This is generally associated with cancers, and we're not going to talk about that here. You'll talk about polycythemias at the very least in pathology in the second year. Okay, and so there are general overall features, and I'll have more to say about this in a second, but there are generally point mutations that can alter structure function. Primarily, we're talking about the hemoglobin S mutation, which is glutamic acid to valine at beta 6. We can also get hemoglobin C, which is glutamic acid to lysine at beta 6. And then since both of these mutations are fairly prominent in the populations that they exist in, primarily in African Americans, but in some other populations, Middle Eastern populations, and some Italian populations, you can inherit both of these together, and so some people walk around with both mutations simultaneously. So they're double heterozygous. They have S and C. In general, there are approximately 1,000 other mutations that are in the literature. You can go to this website, uptodate.com, to look at some of that. You have to register to get in. It's free. I used to have a number of 750 in here, but this continues to go up. Okay, we'll mention a couple of others as we go through. So there are point mutations that affect structure function. There are also mutations that affect either gene deletions or protein levels. So alpha thalassemia, low concentrations of alpha chain, tend to be gene knockouts. They tend to be destruction or partial destruction or loss of alpha genes. Beta thalassemias tend to be impacts on splicing or mRNA processing. Okay, and so again, we're going to get to all of that as we go through. Now that inherited metabolic diseases chapter that I mentioned has a whole series of summaries that I've copy pasted in here as a source of discussion, and you can use this to look at lots more details and learn a lot more about it. They described the inherited disorders of hemoglobin as three overlapping groups. Structural variants, which are the point mutations that I just described. Thalassemias, which are the reduced synthesis of one or more of the globin chains of hemoglobin, the alpha or the beta. And then they say that the third major group is a condition in which fetal hemoglobin synthesis persists beyond the neonatal period. Okay, and this is known as hereditary persistence of fetal hemoglobin. Okay, so this is a state that we're trying to induce in people who have sickle cell disease, but there are some people who walk around with this naturally. And this is basically an effect on the loci that control turning on and turning off fetal hemoglobin. Okay, and so we'll have a little bit more to say about this. This summary says that this is a fairly common mutation in the world, and so they rank it along with the point mutations for sickle cell or hemoglobin C and the thalassemias. Okay, and then they go on to say that there are 750 structural hemoglobins. I think this number correctly now is closer to 1,000. And there it is. And these include hemoglobin S and C. They also mention something called hemoglobin E. Okay, and these mutations may result in chronic hemolytic anemia and vaso-occlusive episodes. That's primarily hemoglobin S. Hemoglobin C is associated with milder hemolytic disease, and hemoglobin E is synthesized at a reduced rate and results in a mild form of beta thalassemia. It's interesting that this mutation is an E converted to a K, so a glutamic acid to a lysine at beta 26. This primarily is an Asian mutation. It primarily exists in Asian populations. It turns out that this mutation is near a splice site. And so this mutation at the DNA level impacts the splicing. And so it's referred to as a cryptic splice site. And the reason that there's reduced amount of protein is sometimes the splicing goes wrong. Okay. There are lots of other mutations that affect stability, folding, or oxygen affinity, et cetera, et cetera. And so a lot of these are things that we'll talk about briefly, but this 1,000 mutations that are out there have other implications for protein hemoglobin structure function. This website that I mentioned lists this also in a slightly different form. They mention single site mutations that affect abnormal polymerization. They affect thalassemia disorders that we've mentioned. They affect solubility of other kinds. So there are some effects on solubility that cause precipitation or crystallization of the protein. There are some that cause folding defects. There are some that affect affinities, either high or low affinities. And there are some that cause met hemoglobin stabilization, which leads to cyanosis that we've talked about in previous lectures. And so this is known as hemoglobin HbM. Okay, and so this is a summary from this website, and it discusses in general features all of the things that I've just said. So I think I've done this sort of 3 times now in terms of the features that we've been talking about. Our primary interest is hemoglobin S or hemoglobin C. These are mutations in the beta chain at beta 6. Hemoglobin S, if inherited from both parents, is what causes disease. Hemoglobin C is this milder form. And then this is pointing out that you can have both mutations, and so you can have hemoglobin SC doubly heterozygous, and this has a completely different phenotype than hemoglobin S or hemoglobin C alone. Okay. This is a slightly different summary from another source that I have shown in the past. I don't think I showed this in the L7, L8 lecture. It's a little bit fuzzy. It's a bad reproduction, but it's a different kind of summary of what we're talking about, examples of hemoglobinopathies. So solubility, decreased, increased oxygen affinity, hemoglobin M, unstable protein, abnormal synthesis, etc. So there's a whole number of features, And you should store this away in terms of structure-function relationships, specifically the kinds of things that might impact normal hemoglobin structure function. It's hard to imagine how effects on oxygen affinity doesn't affect everything else. Okay, and so these things are all coupled to one another in the cooperative nature of hemoglobin, but there are specific mutations that are targeting specific things. And then finally, to kind of end this section of great detail that I've now gone into about the numbers of mutations that are out there, the current status at UMC, I just got these numbers yesterday, actually. So I got these from Dr. Gordon, who some of you will see next week. There are 505 sickle cell disease patients at UMC. There are 292 hemoglobin SC patients, so you see that it's really common. There's a large number of hemoglobin S beta plus mutants, and beta plus means a beta thalassemia, but it's a mild beta thalassemia. There are 89 of those patients. And then there are 26 patients who have S beta naught. Beta naught or beta zero is a complete loss of the beta chain. So there's one healthy chain, one normal gene for beta, but it has a processing error. It doesn't splice correctly. And consequently, those patients are essentially homozygous for S. They only have one copy, but since there's no other beta around, they essentially have hemoglobin S and they have sickle cell disease. There's also a number of other mutations out there. There are some people that we have two patients who have S and D. D is a mutation of a glutamic acid to glutamine at position 121. And there are two of those patients. And then we have one patient who has SE. Remember I said in a previous slide that E is actually an Asian mutation. I think it was E26K. E26K. I should have written that in here. E26K, I think, is what I have up above. Okay. We have one of those. And then we have a number of sickle cell patients. I presume that these patients belong in this 505 group or subgroup, who in addition to having sickle cell disease also have this hereditary persistence of fetal hemoglobin. So these patients will have milder sickle cell because there's such a high concentration of fetal that it dilutes out some of the sickle cell and it diminishes the severity of the disorder, okay? So just like this patient has S beta 0, has worse sickle cell, so they have trait, but they actually have sickle cell disease because they're homozygous for S since there's no B. These patients have milder sickle cell, okay? And it's because it's increased levels of fetal hemoglobin. In general, we don't know how many people in Mississippi or at UMC have trait. That would be SA. They have one normal gene and one abnormal gene. But in general, about 8% of African Americans in the United States carry a single sickle cell gene. Okay, so this is that other paper that I mentioned that's posted out there, and this is sort of a discussion of some history. The sickle cell mutation was first discovered in 1910. A dental student presented with pulmonary symptoms, and this physician, Herrick, coined the term sickle shaped when he looked at the patient's red blood cells, and you could see these abnormally shaped cells. And so this is the first observation of the general features of pain crisis, etc. At the time, it was not clear if this was sui generis, which means a spontaneous mutation in this individual, or whether it was actually inherited. We now know it was inherited. Okay, and so as more and more cases came about, they were able to track that this was actually being inherited from parents, and so that explains what's going on. Okay, it was also around this time noted that when you deny oxygen, these sickle-shaped red blood cells form. In this particular case, apparently, they tied off somebody's finger, and then they did a finger stick later, showed that hypoxia seemed to cause an increase in the proportion of sickle cell shaped cells. And so this is the association with deoxy. This is the association with the T state. So I mean this to be deoxy and the T state. Okay. Okay, and then there's some discussion here down below that we can talk about next week, and you can read in the paper itself. Okay. So generally speaking, we're focusing on sickle cell itself and other mutations that are inherited along with it. So this sickle cell shape was first described by this guy Herrick. It's a genetically transmitted, homozygous recessive, chronic hemolytic disease. So it undergoes Mendelian inheritance. You inherit one gene from your mother and one gene from your father, and if they're both S, you have sickle cell disease. We've mentioned that this is a trait, single copy found in 8 to 10%. This summary that I wrote from some time ago, it's not clear to me what that number really is in the population. I'm not sure that we know. And then the heterozygous form is what we call trait, and the homozygous form is what we call sickle cell disease or sickle cell anemia. I emphasize here that this is a paradigm for molecular disease. There are a number of diseases out there that are clearly associated with a single gene. They're clearly associated with single mutations, okay? And this was first observed for sickle cell. In the late 40s and early 50s, it was hypothesized, I think I say that on another slide, as I often do, that this single mutation, early on it was known that there was a change in charge, and then it eventually was known that it was a glutamic acid valine mutation. That there's a single mutation in a single gene that leads to disease. Okay, and so an inherited disease, and so this is what we mean by a clear molecular disease. Many diseases are multi-genetic, meaning there are multi-genes that are involved, and much, much more complicated. When we get to diabetes, that will be the case. But this is, therefore, falls into a very special category because of this unique feature. Okay. So here's a depiction of the inheritance. If your parents are carriers, okay, normal and abnormal, you have a 50% chance of inheriting each copy from each parent. So you might get the normal copy from both parents. You might get a normal and an abnormal from one parent, or a normal and an abnormal, from another parent, or you might inherit two abnormal copies. Okay, and so this means that 25% of progeny will be normal, 50% of progeny will be carriers, and 25% of progeny will have the disease, inherit both abnormal copies. We've already discussed the fact that you can ascertain in young patients whether they are normal, AA, or whether they carry a fetal copy or sickle cell copy, and whether it's two sickle cell copies or one sickle cell copy. So here's a normal and a sickle cell. This is heterozygous. And whether they have sickle cell A and C. So this is a patient with SC. And then here's a patient who's got S. They must have a thalassemia, because the only other thing that's left is fetal. Okay. And so that's sort of interesting that you would see that. Okay, so this patient is A, and then they seem to have high A, and then they seem to have a high amount of fetal. These other patients don't seem to have high amounts. You can do this by electrophoresis. You can also do this on some kind of chromatography. You discuss with Dr. Hebert, and in your book, Lippincotts, there's a discussion of, you can also do this at the genetic level, and you can do allele-specific oligonucleotide, and you can actually look for mutations and non-mutations by depending on which bands light up in a nucleic acid gel, okay, and then matching with some kind of an allele-specific complementary strand. And then you also can do this by some kind of restriction site mapping, because the mutation in sickle cell actually knocks out a splice site, knocks out a restriction site. And so when the site goes away, you know that that patient has sickle cell, okay? And I think that's depicted down here. This splice site creates a lower molecular weight band. In SS, it's completely gone. In AS, half of the sites are still there. In AA, 100% of the sites are still there. So there's a number of ways, these two techniques and the techniques previously, that you can do this. Now, generally speaking, I have been saying, although not emphasizing, and we will talk about this a little more next week, that people who carry around a single gene that have sickle cell trait generally are asymptomatic. They tend to not have anemia. They tend to not have pain crisis, okay? But this is, in fact, not entirely true. I have two examples here that I'm going to show you. One is this football player, Ryan Clark, who played in the NFL. I don't think he's playing in the NFL any longer. He played for Pittsburgh. And the article is about the fact that he had a sickle cell crisis at a game in Denver, which is at high altitude, so low oxygen. And in a repeat game, the coach was not going to play him because he was worried about his players' health. Okay, so it was a pretty severe state, and you can read the features in this article. Okay, so that was just a big deal at the time, pointing out that this guy had already had a crisis in Denver, and they weren't going to play him in the next game. He was a defensive back, by the way. The game went to overtime. Tim Tebow was the quarterback of Denver at the time. And there was a 80-yard touchdown pass thrown where the defensive back, his substitute, didn't make a tackle, and Denver won the game. Okay. That has nothing to do necessarily with what we're talking about, but here's a situation where somebody with sickle cell trait couldn't play in a game for reasons involving the high altitude conditions in Denver, and it had big consequences in that particular game. I am not wrong in saying it was the best moment of Tim Tebow's short career. He's also not had a very good baseball career, and I think we'll see him on television for the rest of his career. The other example was a few years before that, and many of you may or may not know about this. There was a football player at Ole Miss who collapsed during a practice and died. Okay, so he had sickle cell trait, and this player got extremely dehydrated, lots of exertion. The coroner mentions down here that he also had an enlarged heart, so that might have contributed to the condition. But in the end, this patient with sickle cell trait died in response to exertion and loss of hydration. Okay? And so what's true now in collegiate level sports is that all players, especially in football, are tested, and we know that they are or not have sickle cell trait. And we're very concerned now with features about how they practice and how they're allowed to practice. And there's a lot that is known about that today. As a matter of fact, I'll say one more thing to you. Ole Miss had a women's soccer player, a Brazilian soccer player, a number of years ago. She played on the Brazilian national team for a while. I'm not sure she's still playing for them, but she did for a while. And she also had sickle cell trait. And she basically had a bodyguard. She basically had somebody who followed her around 24/7 because Ole Miss paid a lot of money for this guy because of this guy's death. Okay, and they have put a lot of effort into making sure that anybody in their sports program who has sickle cell trait has the right hydration, is eating properly, is not overexerting, et cetera, et cetera. And I think that's become the common state throughout collegiate sports and probably at pro sports level. I could go on and on about that in other contexts, but we will keep going here. Okay, so general molecular properties of sickle cell. is that it causes the erythrocyte to distort. It increases with deoxygenation, so it's the deoxy form that brings that valine into the proper position that it might aggregate. It is known as the first molecular disease, predicted by Linus Pauling in the late 40s. You can see it in a shift in the charge. This valine is described as a sticky patch that allows tetramers to associate and then aggregate. And those big aggregates distort the shape of the red blood cell, which eventually lead to membrane damage. They lead to red blood cell destruction, so chronic hemolysis, which also then converts or causes inflammation. But the real feature has to do with capillary blocking. Okay, and so here's a cartoon that depicts this sickled shape. You can see a field of a number of sickled cells. The substitution is at this position that allows this kind of an aggregate to form. This image is not appearing. This is basically a cryo image that shows you this structure on its side. Okay. And then this is a slightly different depiction showing where these interactions occur between tetramers leading to this sickling shape. The sickling then causes a blockage of capillaries, which is basically pain throughout the body. And so those are the kinds of things that we'll be talking about in the clinical next week. Okay. And so there are lots of different features of tissue anoxia resulting in severe pain, including migraines, chest pain, hand and foot pain, et cetera, et cetera. So those are the features that you need to be investigating on your own. Okay. The description of the sickling is described as a solubility. Okay, so deoxyhemoglobin S is 25 times less soluble than deoxyhemoglobin A. It's a sticky patch that interacts with this leucine at position 88 that forms the stability of that 14-stranded polymer. Okay. And so this 14-stranded polymer will form under conditions of low oxygen. So that means in the capillaries where the oxygen concentration is dropped to 20 and below. Okay. Consequently, the sickling is really a kinetic process that depends upon the time that the red blood cells spend in this low oxygen environment. And it also depends upon concentration because it's an aggregation and it's concentration driven. So how much hemoglobin S do you have? How much fetal do you have? Do you have any fetal in A? This will dilute out the S, and that's why trait patients generally don't have this problem. And then you can have other mutations that could also impact this. Okay. It causes membrane damage, cation release, hemoglobin denaturation, cell fragmentation. It's a hemolytic anemia because the red blood cells are destroyed. Increased intracellular viscosity, occlusion of microvascular, which leads to pain, ischemia in tissues. It can lead to enlarged heart. Remember that football player from Ole Miss had an enlarged heart. It will lead to inflammation and bacterial infections. And in some sickle cell patients, for some reason, they end up with higher persistent fetal hemoglobin expression, which is actually ameliorating, which actually reduces a lot of these phenomena. Okay. So this slide is repeating a lot of the things that I've already said. Sickle cell, red blood cell, sickling of red blood cells in SS patients begins below about 80 torr, 80 in terms of partial pressure of oxygen. And then you can get significant sickling below 60 and certainly down to 20. Heterozygous sickle cell will generally, so trait will generally not begin to sickle until it's below 20. Remember I said that it's a kinetic phenomena, and so it transported into conditions where there's high oxygen, and so it generally doesn't happen. Okay? And so this is basically restating a lot of the things that I've already said. So generally speaking, For all the reasons that I've mentioned, SS will generally sickle and AS will generally not sickle unless you're dehydrated and playing football at high altitude in Denver. Okay. And so this is simply emphasizing down here is where we're talking about the preponderance of the sickling condition, and SS patients will have that. Okay. There are other things that can compound this. So heart disease, chronic lung disease, which means low oxygen exposure to high altitude, exertion and dehydration, bacterial infection. In the case next week, some of these features come up as part of the discussion. Okay. So why does this mutation exist at such a high level in African populations, or in Mediterranean populations, or in Southern Italian populations? This falls into the general category known as balanced polymorphism. It turns out that this region of Africa tends to have a high population of malaria. Okay, so this is where we see malaria in Africa. You can see some up here in the Mediterranean, but mostly it's in Central Africa. And then here is where this mutation tends to exist in Africa. And these are numbers in the population. So you can see in some African populations, as many as 20% of the population carries one copy of the gene. Okay? And that's because hemoglobin S provides some selective protection against malaria. The general life cycle of the malaria organism is in red blood cells. So to develop, it needs intact red blood cells. And hemoglobin S patients tend to have less fragile, or more fragile, less stable red blood cells. And so consequently, the life cycle of the malaria organism is reduced. Okay, and so the malaria isn't as effective in those people who carry one copy. Okay, it reduces slightly the red blood cell half-life. Okay, it prevents parasite development. There's some discussion that the release of potassium is involved in this ability to kill this organism. I think that's a little bit uncertain and controversial, but it's the way we talk about it. When red blood cells break open, their ionic content is broken up or released into the bloodstream. Okay, but it's this protective effect of hemoglobin S that seems to explain why this is prominent in this population. These people tend to survive malaria exposure, and so they become enriched in that population. Okay. SS patients, who are a necessary consequence of this, don't necessarily survive because they might die young of pain crisis, of all of the other features that we talk about in sickle cell disease patients. But it's the trait patients who really lead to this condition. Okay, and so it turns out that this phenomena is not just a sickle cell disease in Central Africa. It turns out that provinces in Saudi Arabia and people in India also have mild sickling disorders and higher levels of hemoglobin F that are associated with factors that prevent problems with malaria or protect them against problems with malaria. Okay, and so the prognostic factors having to do with high levels of other mutations and how they might change the phenotypes or the way the penetrance of patients with sickle cell disease is observed in lots of other regions in the world. Okay, and I'll have more to say about that in a second. It was noticed that especially this hemoglobin F connection, we established early on in the 80s, 70s, and 80s that this had a protective effect. And so early on, it was hypothesized that one way of treating sickle cell is to increase artificially hemoglobin F through DNA modifications, through methylations and acetylation reactions. And that's when we started looking for hemoglobin F inducers, and that's when the whole hydroxyurea study began. There are lots of other things that you can look at, and this is all discussed in that other chapter from that article from 2007. And you can read about all of those features. All of these have been in clinical trials, and it's the hydroxyurea that we seem to have settled on for a lot of the treatment studies. Okay, so now let's switch to gene deletions and RNA processing. So again, you have 4 copies total of alpha, and you have 2 copies total of beta because we're diploid. So this is the gene structure on a single DNA strand, but we are diploid, so we have 2 strands. Okay, and again, this lists lots of the genetic modifications that can occur that might affect hemoglobin expression. So there are single-site mutations. There are some examples of 2-site mutations. There are some weird examples of fusion mutations, clearly deletions or insertion mutations, okay? There are all kinds of genetic features that exist in that 1,000 mutation examples that I mentioned from this website, okay? But primarily what we observe in the African American population, especially in a place like Jackson, in Mississippi, as I mentioned up above, are thalassemias. They're hereditary hemolytic diseases in which there's an imbalance in the synthesis of global chains. So again, normal hemoglobin is alpha 2, beta 2. And the assumption is that these 2 chains get expressed, synthesized in equal amounts so that you can make tetramers. And if there's an imbalance in the amount of alpha or the imbalance in the amount of beta, then you can't form normal tetramers, okay? All right? And so you can't form normal amounts of hemoglobin A, okay? And this can be due to gene deletions of several nucleotides, or it can be due to nucleotide mutations that affect synthesis and processing. The general nomenclature is that if you have complete loss of alpha or beta, they're called beta naught or beta 0, okay? So you can have alpha 0 thalassemia or beta 0 thalassemia, okay? Or if there's partial reduction, then it's called alpha plus or beta plus thalassemia, reduced synthesis, due either to gene knockout or reduced processing, okay? So that's the nomenclature you should be familiar with. This is a summary from that inherited disorders chapter. This at the time is identifying 170 mutations identified as causing beta thalassemia. Mostly they affect transcription or processing or translation. Some of them have to do with unstable chains, so that's a folding mutation. And then the common form of alpha thalassemias, as I keep saying to you, are deletions that involving one or both of the linked alpha globin genes. So there's 2 copies on each DNA, and generally when you delete them, you can delete one or both of them. You don't tend to delete them across the DNA chain, set in different chains. You tend to delete them on the same chain. Okay? And so that's the summary that you can see in words. So what's interesting is that these thalassemias exist in Africa, in the same regions in Africa that the S mutations are. But as I'll show you below, they're actually more prevalent in Asia. Okay, and so here's a summary of the splicing defects that might occur due to mutations that affect splicing and therefore affect stability of beta chains primarily. Okay, and so you could look at that in this chapter. So here's the summary of the alpha thalassemia mutations. You'll notice that they occur in Africa, they occur in the Middle East, they occur in Southern Europe, okay, but they exist predominantly, you can see the blackness of some of these, in places like India, but also in places like Thailand and Indonesia, okay? And so they're primarily Asian mutations. And then this figure summarizes what those mutations are and what codons there are where in the genes they exist. Okay, and so these also happen to be malaria regions. Okay, and these malaria regions also respond to anemias. So survival is coupled to anemias, the life cycle of the malaria organisms. And so these mild alpha thalassemias also have some kind of a protective effect, and that's why they're found in increased concentrations in that population. Okay, so this is a summary that comes out of Lippincott, and this is a summary of what happens in various kinds of thalassemias. Okay, so the key here is that we have 4 total copies of alpha, and we can lose 1 total gene of alpha, and it would be known as a silent carrier because we still have 3 copies. And then we can lose 2 copies, either from different chains or along the same chain. And these begin to show mild thalassemias, mild anemias, okay? And these tend to be mild forms, okay? But you can then lose 3 copies. And when you lose 3 copies of alpha, you begin to have significant reduction in alpha chain expression. Okay, this is actually known as hemoglobin H. And when you have reduced amounts of alpha, you can't make normal amounts of tetramers. And so you have excess amounts of beta, and you have excess amounts of fetal. Okay, so the fetal excess will tend to form if that responds to the alpha thalassemia. So the fetal form will tend to make a tetramer that is not cooperative in oxygen binding. It's known as hemoglobin Barts. It is dysfunctional, essentially. And then the beta chains can also make tetramers. So these would be beta 4s. These are known as hemoglobin H's. They do not bind oxygen cooperatively. They do not deliver oxygen. And then a lot of the beta chains simply precipitate. They're insoluble. Okay. So as you get rid of more and more alpha chains, you begin to develop these other forms that end up being dysfunctional and toxic. So once you lose all the alphas, this basically is fatal. Hydrops fetalia, you cannot survive. So you're born, the baby had survived it up to that point with tetramers made of some alpha and delta, but generally speaking, they're already sick and they'll die soon after birth. Alternatively, in the beta thalassemia, you can lose 1 beta chain expression, and you can have reduced amount. This is minor, so this would be the beta plus. This would be beta plus, or you can have complete loss, and this would be beta 0. Okay? So in this case, you have reduced amounts of beta chain. Again, you can't make normal tetramers. You again will form tetramers made of just fetal hemoglobin Barts. And then you will tend to form tetramers made of the alpha and the fetal forms. This is known as hemoglobin F. It also doesn't function very well in terms of oxygen binding. And then the excess alpha chains will also tend to precipitate. Okay? So the severity of your disease, whether it's a beta thalassemia or an alpha thalassemia, will depend upon what the expression levels are, okay? And how well your oxygen transport systems are working, and at what point it causes severe disease. And the feature about that then becomes more interesting is that you can inherit these along with things like sickle cell. And so when you inherit these along with sickle cell, or you can inherit these along with hemoglobin C, then the phenotype is different again, okay? Because you're double heterozygous, and this will also introduce unusual, complicated pathology, okay? And so understanding how all of these evolve and what impact they might have on oxygen transport, oxygen, hemoglobin structure, etc., are important. But you can see from our notation up above at UMC that many patients that we have both They have both sickle cell and some kind of a thalassemia. Okay. And so this is from that inherited chapter that I've already mentioned to you. And this is showing some examples of different combinations and what the form of the severity of their anemia is, what the genes are, what their general expression patterns are, and how that expression pattern might impact the disease. And notice in these particular examples, there's generally no dramatic increase in F. It's simply a combination of there's a little bit of A or there's no A in combination with other types of mutations. Okay. So the final thing we're going to discuss has to do with what we're trying to do about this. There are ways to impact this that involve external sources, drugs, for example, that might increase the amount of fetal expression. That works somewhat in many patients, but it doesn't necessarily work in all patients, as we'll discuss. But generally speaking, the main emphasis these days is on trying to change this genetically, trying to, in some genetic way, increase fetal expression, or actually go in and fix the gene problem, actually go in and substitute normal hemoglobin A for hemoglobin S in some modification. Okay, and so we'll discuss transplantation, bone marrow transplantation. This is generally a dangerous way to go, but a lot of this is now what's being investigated. And this entire discussion kind of summarizes that entire sort of investigation, including drugs and then mutagenic sort of approaches. Okay, and so both of these lists of words that you have here should be used to sort of bring you up to speed with that. The genetic theory that's been going on is kind of accelerated recently. There have been lots of attempts to increase fetal hemoglobin primarily by affecting the locus that turns off hemoglobin F. So that's one of the primary things that have been going on. Another thing that's been going on recently is to try to put in a normal hemoglobin A gene into the bone marrow. This has primarily been going on since the early 2010s to currently, and there's some success in individual patients. And then recently, there's a specific emphasis on CRISPR-Cas9 because it's a much more targeted approach. I think Dr. Hebert covered a Mississippi patient who has recently had this treatment. They're going in her case, and trying to increase fetal expression by mutating what I discussed up here, this locus that turns off fetal hemoglobin. And I believe she is doing well since her treatment. So they're trying to turn on the gene for fetal hemoglobin. So there's lots of things that are going on and lots of different kinds of trials. I'm just going to discuss one of these. This is a recent New England Journal of Medicine article where they put in a normal hemoglobin into a patient who was sickle cell. So this patient had no normal hemoglobin, and he was basically surviving on blood transfusions. Apparently, he had very severe hemoglobin S. So this hemoglobin A that he was showing here in his blood was because he was living on transfusions. And they managed to put a modification in, an unusual modified hemoglobin A, because it has anti-sickling properties. Not only does it not participate, it has an inhibiting characteristic. And they managed to get this gene copied into the bone marrow of this patient and expressing normal levels of hemoglobin A with anti-sickling properties, reduce the amount of hemoglobin S that was being expressed. And this patient over the short term has been doing extremely well. They're having no more pain crisis. They're having no more episodes. And this has worked. Okay. So this is a successful example of one. Okay. This is a French study, actually. And these people have been working with this particular form for a number of years, putting it into a number of patients. Previous work described examples that failed for various reasons, that the patients didn't take up the gene and weren't expressing it in their bone marrow. Okay. So that's what I have to say. We're exactly at 60 minutes since I started. So this is a summary that comes out of your Lippincott synthesis of structural abnormal hemoglobins is described here, and the various features of S and C and SC. Insufficient quantities, the thalassemias for alpha, thalassemias for beta, the various forms that exist, the hemoglobin SPART, hemoglobin H, and the hemoglobin F. That's in your summaries. And then there's some discussion of methemoglobins. There are some mutations that lead to this. There are some drugs that lead to this. There's some cyanosis that occurs in terms of diagnosing this. And so these are the various things in an overview that we have talked about. We talked about a lot more examples that exist out there. And so you should use this as a general summary, okay? I'm going to post this PowerPoint online. In addition, I have a whole series of other slides that come from this website. They're primarily inheritance patterns, and some of them have to do with the features that I described in terms of hydration treatment, etc. You can use these for your own devices, okay? So I'm going to stop with that. This is as much as you should use for your preparation, and I will see you next Thursday. Okay, and so I'm going to come up here and click end the meeting, and I will talk to you next week. I am going to post this as an MP4, and I will also then post a PowerPoint. So you'll be able to go through this as a lecture, and then you'll also have access to the PowerPoint. Talk to you next week.